All right, so my name's Luke. I'm a postdoc in Jonathan Weissman's lab, who will be here later this week. So I'm giving two lectures, the lecture right now and then tomorrow morning, and filling in for Jonathan for one of those. I'm not sure which one. So I'm a poor man's Jonathan, but that's all you've got. There's going to be a sort of a number of lectures at this point where we depart from genome engineering and talk about other applications of CRISPR. And this will include both of my lectures as well as, in part, I think, some of the work that Martin will talk about in relation to screening. I've not really split my lectures into two defined sections. I have a series of slides, so we'll see how far we get today, and then I'll adjust my slides for tomorrow to proceed accordingly. So I'm going to talk generally about repurposing CRISPR as an RNA-guided DNA binding platform and what we've done with this. A lot of this work has been done at UCSF, and that's why I'm talking about this. But I'll highlight work from Boston and elsewhere, and I think hopefully keep you guys entertained. So what am I going to talk about over the next 2.5 hours? So, I'm going to start with a brief introduction. I'm going to use this introduction to try to motivate why we've done some of the experiments we've done and where we're interested in going with this. Then I'll talk about, so as part of this introduction, I'm going to talk a little bit about programmable DNA binding proteins in general and motivate how we've been able to move quickly over the last couple of years by learning from past work defining how transcription factors work and then creating artificial transcription factors over the last 20 years or so. I'll tell you about how we repurposed CRISPR as an RNA-guided DNA binding platform. And then I'll tell you sort of in a lot of detail about the applications that we've created using dCas9, including using dCas9 to turn genes on and off, to edit the epigenome, to image the genome in living cells, or to image or edit the transcriptome, and then more recently as well as a new platform for making more defined mutations to the genome that don't rely on double-strand breaks. So that's the plan. So how do we get into this, though? I think we and everybody else in the whole world seemingly were interested in Jennifer's paper in August 2012, which described the possibility of easy-to-use RNA-guided genome editing or DNA editing. And so this idea of programmable DNA editing is not the newest idea. People have been using zinc fingers and TALENs for quite a few years. But the problem with these previous platforms was always that you had to design a new protein for every target sequence. And so most labs were a little hesitant to get into this because it took some technical know-how and was not a trivial amount of work. And Jennifer's paper came out and just changed, I think, people's minds on how easy this could be to do. And Dana's talked about the sort of explosion in applications in a variety of organisms and labs across the world. So at UCSF, we had a slightly different take on this in that we wondered from the original work what would happen if you actually broke Cas9 and removed both of its nuclease domains. So in Jennifer's original paper, they did an experiment which is shown here in which they mutated, as Dana probably told you, there's two nuclease domains. There's the RevC and the HNH nuclease domains. Martin in Jennifer's lab showed that you could mutate one or the other and produce a nickase. And so this has applications for genome engineering, like Dana just talked about. What this experiment shows, at least in vitro, is that you can make mutations to the nuclease domains that do not destroy Cas9's ability to bind nucleic acids in an sgRNA sequence-specific manner. And so what we wondered is what happens if you actually just go ahead and break the protein completely and divorce it from its natural function and make a nuclease inactive version of Cas9. So Jonathan likes to describe this as a form of breaking these molecular scissors. And so initially this was really the work of Stanley Shi, who was a fellow at UCSF, now with his own lab at Stanford. And Stanley got into this because he was actually a grad student here at Berkeley with in Adam Arkin's lab, but was sort of jointly advised by Jennifer, and so was in on the Cas9 revolution from the start. And Stanley, being a bioengineer, thinks in ways that sometimes surprise me, and he's very creative. And so he wondered, can you make a nuclease deactivated version of Cas9? And if you can make a nuclease deactivated version of Cas9 that still binds to DNA in a sequence-specific sgRNA-dependent manner, what could you do with this? And so this really, to restate it, has been an ongoing series of experiments to define what we can do with an RNA-guided DNA binding platform. And so I think what we were cautiously wondering from the start is, can we create a modified form of CRISPR in which we have an RNA-guided DNA binding platform such that we can, in theory, deliver any protein to an arbitrary sequence of DNA in any cell? And so I think this general question has implications for a lot of fields, including anything with a nucleus or anything with DNA, and asking how that DNA is regulated or governed during cell division or the function of any cell. This is sort of too general of a question to ask, though, right? Like, how do you start to ask what you can do with a protein that can bind DNA in any cell? And so what instead we've done, both in E. coli, yeast, and human cells, is ask instead whether we can use this to just turn genes on and off initially. So can we use a nuclease deactivated version of Cas9 to turn genes on and off? And this sort of focused us and gave us a more limited set of experimental goals. And we started with this application of dCas9 largely because we had, in part, a template from other platforms to work from. And so that's just restated here. Can we create a synthetic tool set that enables us to robustly and specifically turn genes on and off in any cell? So I want to take a step back and though ask, why would we even want to do this in the first place? If you can just use Cas9 as a nuclease to delete genes, what do you care if you can turn genes on and off? And I think the motivation for this stems in part from just a very, very basic observation. So we all have many cell types in our body, but we have the same genome in each of these cell types. And so this clearly demonstrates, I think, that throughout all life, there are dynamic processes that occur independent of the sequence of the genome of an organism. And so it would be useful for us to understand a lot of types of biology if we could specifically mutate the expression of genes encoded in our genome without actually changing the genome in a robust way. And so another way to think about this is it's not just, sometimes we like to think about this sort of like a piano. It's not just that you have a defined set of keys that when struck have a certain sound associated with striking that key, but rather the composition of an organism or a specific cell type relates more to the intensity with which you play those keys and the coordination of playing those keys. So it's not just how many keys or how many genes do we have, but it's how do we orchestrate the expression of these genes across different cell types or processes. Another way to state this is we know in the human body we have like at least 200 cell types. They all have the same genome, but they have very different functions. So you have neurons, you have T cells, you have muscle cells. They all have the same genome. They all serve very different functions, all of which we're very appreciative of. But how do these cells know during development and during normal tissue homeostasis how much of each gene to express? And how does the expression of each gene relate to the function of a cell? So I think these are very, very broad questions that don't relate to any like one disease or any one specific process. But if we had tools with which we could dynamically control the expression of individual genes and ask how turning up or down or off individual genes affects the function or output of a cell, this would be broadly useful in neurobiology and developmental biology and et cetera. Okay, so I'm going to take one step back, though, even further and talk about transcription just very generally. So I've been dancing around the idea that transcription controls the expression of genes. Transcription is the process by which the information encoded in your genome is turned into RNA molecules, which are then translated into proteins. So this is, perhaps much too basic a slide for this kind of a workshop, but I'd just like to orient everybody on the same page to the fact that this is been described as the central dogma in biology, and what Dana's described has all been at the level of manipulating the genome. But there are other ways you can manipulate cells. So you can manipulate transcription, or you can manipulate translation, and all of these in the end can have similar effects on modifying the output of a cell or the function of a cell, either by deleting genes, by controlling transcription, or by controlling translation. And so if the first series of lectures were about DNA, the next series, or my entire talk, is going to be more or less about modulating transcription and how this can teach us new things about how cells function. Okay, so what is transcription? Transcription is a process by which genetic information encoded in the genome in your genes is turned into RNA molecules. There are two things that have to happen to turn DNA information into RNA. One is that you need general transcription machinery that is RNA polymerase that actually produces a specific mRNA, but you also need sequence-specific transcription factors. So I won't talk much about general transcription machinery. This is conserved machinery that exists across all life, with additional factors present in eukaryotes that help transcribe longer genes. But I think what is more relevant to today's lecture, and what is, I think, sort of a bigger ongoing question is how does every cell that exists know how much of each gene to make and when to make it? So this is very simple. This is sort of hardwired into things. But the bigger question is, how do you regulate things? So I think a very basic observation that that was influential in my thinking about this comes from just looking at different species and asking how big is their genome, how many genes are encoded, and how many sequence-specific transcription factors do they have. All of these organisms have a basic RNA polymerase, but they have very unequal distribution of sequence-specific transcription factors that governs how dynamically they regulate their genome. So let's just use three organisms as an example. This is a mycobacterium that has one of the simplest genomes, or was one of the simplest forms of life that we know of. This was the bacteria that was used by Craig Ventnor to try to create a synthetic bacteria as the first synthetic form of life. So this bacteria has a genome that's only less than 1 million base pairs, 580,000 base pairs. It only encodes 517 genes, and there are no sequence-specific transcription factors encoded in this bacteria. So this is basically the simplest form of life, where there are genes that are expressed at different levels, but as far as we think about, conventionally, there's no specific regulation of these genes. So this is as simple as it gets. Yeast are a workhorse organism, or have been for a long time. They represent sort of an intermediate level of complexity in terms of life. They have a genome of, 12.5 million base pairs with about 5,770 genes, so about tenfold more roughly than this guy. And here now you see that yeast respond dynamically to a lot of processes, and to govern this, they've now evolved about 169 transcription factors, give or take a few, depending on how you define this. And so these transcription factors really, and I'll talk a little bit more about this, give yeast the ability to dynamically regulate their cell cycle, whether they're going through sporulation and other things, meiosis, or as well as dynamically regulate their utilization of different carbon sources and or during fermentation. And most of these dynamic behaviors have inputs that are dependent on a lot of proteins, but the most downstream output that controls these dynamic lifestyle changes are transcription factors that fundamentally alter the way a cell behavior is governed between different states that a cell is trying to move towards. So, and then because humans are human-centric, I'll just use humans as an example of metazoans. We have about 200 cell types in our body. Our genome size is just a little bit over 3 billion base pairs. We have, about roughly, depending on how you define it and whether you include non-coding transcripts, we have about, this is excluding non-coding transcripts. We have about 21,000 protein coding genes, so about 4-fold more than yeast. But now you see that we have about a 10-fold increase in the number of sequence-specific transcription factors. And this increase in transcription factors is one of the major things that drives our ability to go from 1 cell type to 200 cell types. And I'll talk more about a couple classes of transcription factors, but one of the fastest evolving sets of genes that differentiates, apes and monkeys from humans is several specific classes of transcription factors. So these are very clearly important for the way our brains work differently from an ape's brains, or our ability to evolve as humans. So the point here is that gene expression is complicated, and I'll reiterate this a couple more times in the next couple slides. It's controlled by multiple factors. Transcription factors clearly have evolved to be able to allow us to dynamically regulate gene expression. But then there's other layers of organization that are also important beyond just simple sequence-specific transcription factors. And what I hope to convince you of by the end of the next 2 lectures is that we can modulate potentially all of these with dCas9. So this is sort of a general motivation slide that just is to put everybody on the same page. Transcription in human cells is complicated. It requires an interplay between different general transcription factors, activators, repressors, and co-activators. There are lots of feedback loops that keep things on or off or limit the length of expression. And there are lots of regulatory sequences both next to transcription start sites of genes and very distal in the form of promoters and enhancers that can influence gene expression. And then lastly, this picture shows, I think this is a B cell. And our genome does not exist just as DNA, but rather it is highly chromatinized, and this chromatin structure governs, the epigenetic code governs both gene expression and nuclear architecture, all of which influence gene expression. So I'm not going to go through this whole slide. Basically, the point is that transcription is governed by sets of proteins, and these proteins are highly integrated, and there's a lot of them that are responsible for correctly expressing genes across development or across the different cell types encoded in our body. And it's not, as I said, just DNA. There's also, this is a complex interplay of lots of proteins, some of which are structural, like histones, and we'll talk more about this later. Okay, so hopefully I've convinced you that transcription is complicated. In the early 80s, people had decided that they knew enough to know that we didn't know anything about transcription. And so people decided to use model organisms to try to ask very, very simple questions about how 1 or 2 genes were expressed. And these seminal experiments really led to a huge amount of groundwork that still holds true about how transcription works and how genes are turned on and off. So I'll go through just a very small, just 4 or 5 slides defining how we figured out, or how people in biology figured out how transcription works in E. coli and yeast, and then talk about how that led directly to the creation of artificial transcription factors that we're still more or less using today. Okay, so transcription factors are modular proteins. Their defining feature is that they have a DNA binding domain that has some sequence-specific recognition. You can get transcription factors that act as repressors or activators. They often act as scaffolds to recruit additional domains. And a key observation that I'll highlight on the next couple slides is that you can usually split the DNA binding activity from the effector activity. And this is crucial for thinking about artificial transcription factors. So, like I said, in the 80s, people realized that we were interested in transcription and that we didn't know anything about it. And so there's this classic series of experiments in E. coli looking at the lambda phage. It was known that lambda has two different life cycles. There's a lytic cycle and a lysogenic cycle. And people knew that there was essentially one protein that governed the switch between lytic and lysogenic phage activity. And they knew the binding sites, and this led them to what I think is one of the first really concrete questions about transcription that gave us an insight into how transcription factors work. So they had this protein that binds to these two domains. When it's bound, let's get this backwards. This lytic gene is turned off, and this lysogenic gene is turned on. So they knew it bound, they knew it controlled the activity of two different genes in opposite orientations, and this led them to ask a question about what this protein does. This is presented as two different models. So model one was that this DNA binding protein binds DNA in a way that stabilizes a DNA structure that allows the recruitment of general transcription factors such as RNA polymerase, keeping this gene on and this gene off in some kind of a weird structural orientation. Model 2 is sort of more simple and more, in my mind, more elegant in that they hypothesized that perhaps this protein binds DNA in a sequence-specific manner and recruits additional proteins that then turn this gene off and in some way keep this gene turn this gene on and in some way turn this gene off. And so they went about to try to ask which of these hypotheses were true. And to do that, they just mutagenized the lambda and looked for mutants where they could dissociate DNA binding from the protein function. So they found one of these mutants where the protein still bound to DNA, and this gene stayed off, but now this gene was unable to be turned on. And so this suggested that DNA binding activity could be dissociated from the activity that was required for turning this gene on, and in some ways was some of the first evidence that sequence-specific transcription factors recruit additional proteins to turn genes on or off. But phage are pretty simple, and people weren't sure that this was going to be true in eukaryotes, and so the sort of debate raged on for a couple more years. People switched to using yeast to try to answer this question about whether sequence-specific transcription factors stabilize DNA structures or whether they recruit protein domains to turn genes on and off. And there's a lot of, so I'm glossing over a lot of people's work here, including people here at Berkeley. I'm just talking about a couple key experiments, but a lot of the work that was done looked at carbon utilization in yeast, at least for figuring out this in eukaryotes. And so for the next couple slides, I'm going to talk about the Gal1, Gal4 systems in yeast. In yeast, it was known at this point, sorry about that. It was known at this point that if you grew yeast in glucose, Gal1 transcription is turned off, dependent on a DNA binding sequence termed an upstream element in a Gal4 and Gal80 dependent fashion. If you switched yeast from a glucose carbon source to a galactose carbon source, people knew that Gal1 transcription would go on and that you maintained Gal4 binding, but that Gal80 would dissociate. But they didn't really know. This was a descriptive series of observations. They didn't really know whether this was due to some specific recruitment of some structure or what. And so there were essentially 3 experiments that answered this question, and these experiments also gave us insight into how transcription factors work that allowed us to create artificial transcription factors. So this work was largely done by Mark Ptashny's lab, or at least the experiments I'll describe here. And so they knew this much about the system, but they wanted to know why these observations were true. And so they decided that the easiest way to test this was just to make a series of alterations to the Gal4 protein in which they would try to ask whether they could dissociate DNA binding activity of Gal4 from its ability to activate or inactivate gene expression. And this was done in the mid-'80s, sort of at the cusp of when people just after people had invented, restriction enzymes and things like this, and were starting to create defined mutations to plasmids that would allow them to ask questions about gene function. So these experiments seem very simple, but honestly, just creating a couple deletion mutations was enough for a science paper. So that's things have changed. This is the experiment they did. They took Gal4 and they deleted chunks off the end or chunks out of the middle and just asked, do you still get binding to DNA, and do you still activate transcription? And what they were able to show was that there are a variety of mutations where you do get DNA binding. So this is they defined this as the DNA binding portion of the protein, but where you do not get activation of transcription. And you can also use, having mapped this DNA binding domain, use additional internal deletion mutants to show that most of the protein is not required for either DNA binding or for activating transcription, but there are just these 2 minimal domains at the N and C terminus that one is responsible for DNA sequence recognition, and the other is responsible for turning genes on. And that's it. That was the first set of experiments that defined in a sort of controlled way how transcription factors work, and showed that there are specific domains that bind DNA and there are specific domains that recruit effector domains that control transcription, leading to this model where you have DNA binding domains and you have regulatory domains. This is a cartoon from a review, but I'd like to. This summarizes all the information you really need to know. But what I think is funny is that this is the actual data. So it looks exactly like their cartoon. These are just self-reported beta-galactosidase numbers with cartoons. That's it. That's all they showed in the paper. So those series of experiments defined that transcription factors were modular, that they were composed of DNA binding domains and domains that either activated or repressed genes in a specific way by recruiting additional proteins. And the next, I think, fairly obvious question at the time from the same lab was, okay, can we move beyond this and further prove this by creating an artificial system in which we graft either the DNA binding domain or the activity domain of specific proteins onto a new protein and create chimeric proteins in which we can dissociate the sequence specificity from the activity of a protein. And so to go about doing this, this lab created the very first artificial transcription factor that I'm aware of, in which they fused the Gal4 yeast protein. It's Gal4 yeast protein without the DNA binding domain to the DNA binding domain of another bacterial transcription factor that they knew about, this protein called LEX-A. And then they asked whether Gal4 was still regulated by the carbon source input, or whether they could artificially force transcription of a series of plasmid reporters by recruiting the activation domain of Gal4 to DNA using the LEX-A DNA binding domain. And so that's shown here with these series of reporters. I'm not going to go through all of it. Basically, this table is the results from the whole paper. And what they were able to show is that these are all controls, essentially, that their plasmids work as expected, that things are off in the presence of galactose and other way around, on in the presence of galactose and off in glucose. And then the point of the paper is actually just this, that you can force transcription to stay on if you express this fusion protein in the presence of glucose. And this was the first experiment that showed that these kind of artificial transcription factors would work, and that you could couple the DNA binding specificity of one protein to the activity of another protein and force transcription to do what you want it to do. So this is yeast are also, fairly simple. People quickly asked whether this would work in human cells, and they fused these same Gal4 proteins to activation domains that activate transcription in human cells, so to the ER activation domains or the glucocorticoid receptor activation domains, and then asked using reporters whether they could turn gene expression on by recruiting domains to specific regions of this reporter in a Gal4 dependent way. And we're able to show that yes. So like here, for example, you have this reporter where you have Gal4 binding sequences, and it's off until you add estrogen or tamoxifen, at which point the transcription goes on. Same here. And I think what's important here is that these early papers defined activation domains that we're still using. So this is the first Gal4 VP16 fusion. People define that there are domains in the herpes virus that are really potent activators of human gene expression, and this is the domain that we'll talk more about throughout the talk. But this is a domain that we're still all using. I think this work was extended and has been used most broadly, perhaps in the context of TET on and TET off systems. So likely many of you use TET on and TET off systems for inducible or regulatable gene expression in your experiments. This was the next step in artificial transcription factor research, where people started to make more and more sophisticated inducible versions of artificial transcription factors to control gene expression. And the one that I think probably we're still mostly using are these TET on and TET off systems. So this is from '92 from a group where they showed that you could fuse the TET-R bacterial transcription factor to this VP16 domain, and then in the depending on whether you're talking about a TET on or a TET off system, in this case this is a TET off system. In the presence of doxycycline, transcription is robustly driven from this promoter, so this is a luciferase assay. And then if you withdraw doxycycline, you see that transcription turns off because the TET protein no longer binds to the promoter. At the same time, people started to ask whether they could use artificial transcription factors to recruit other activities beyond just activating gene expression to reporters. And so they started to make a variety of Gal4 or TET-R fusions where they fused on putative repressor proteins. And so this is one of the first implementations where this lab fused a series of crab domains to the Gal4 and showed that you could turn gene expression off with these artificial transcription factors. And so this is, at this point, sort of in the mid-90s, we had a series of tools where we had artificial transcription factors that could bind to specific DNA sequences, and you could programmably either turn gene expression on or off by appending either an activation domain or a repression domain to a fixed sequence transcription factor. And there was a sort of expanding tool chest of transcription factors that could be used, such as LEX-A and Gal4 and TET-R, but all of these were limited to their intrinsic sequence specificity. So you had to knock in a TET-R binding site or a Gal4 binding site or a LEX-A binding site into the genome, or you had to express a plasmid that had these binding sites in order to turn on or off gene expression. And there was, at this point, no way to turn on or off endogenous gene expression. And so the next, two more slides on this. People still use fixed sequence artificial transcription factors with increasing complexity. So this is a paper from 2012 in which they knocked in binding sites for this zinc finger protein or Gal4 into a region just upstream of the OCT4 locus in mouse ES cells, and then they made a mouse where now in an inducible way by either adding rapamycin or abscisic acid, you can recruit either this potent silencing domain or a potent activation domain upstream of OCT4, and then ask questions such as how these factors recruit either basal transcription machinery or silencing machinery. You can ask questions about how the order of recruitment of different domains controls transcription. You can ask questions about co-occupancy and who's a winner. If you recruit two potent repressor and a potent activator at the same time, what happens? Or you can do things like order of addition. So they were especially interested in studying heterochromatin formation and maintenance over time, and used these sort of sequential order of addition experiments to suss out how heterochromatin is maintained at specific loci during either differentiation or development. Moving beyond this, a lot of people are still using these fixed sequence transcription factors to inducibly either label or control transcription in the brain. So this is a nice example from 2013 in which they took things even a step further and have an artificial transcription factor that doesn't actually recruit an effector domain, but instead binds GFP. So they had transcription factors fused to single chain variable fragments that recognize GFP, and then they're using GFP as a scaffold to recruit different effector domains or CRI or a variety of different things. And then they label different neural populations and ask about questions about how turning on or off specific genes affects the function of these specific neurons. So these are sort of the current day evolution of the very old experiments I told you about previously, but just to emphasize that these are still very effective tools. They are very well characterized, and they're still useful in inventive ways. But the next evolution of artificial transcription factor research was really to move beyond fixed sequence transcription factors towards artificial transcription factors that have an evolvable or programmable DNA binding capacity. And so the first implementation of this was the zinc finger proteins. And the zinc finger, Dana knows way more about zinc finger proteins than me, but zinc finger proteins have an evolvable capacity for recognizing specific sequences of information. So you can create either in a designed way or in a selection experiment artificial zinc finger proteins that recognize specific sequences of DNA, and then append, once you've found your specific zinc finger that recognizes your desired target, append a variety of domains to control the expression of endogenous genes, and in theory, control the expression of any endogenous genes. So that data is shown here where people turned on off or on expression of just I think one endogenous gene by first defining a zinc finger that binds efficiently to a sequence in the promoter of this endogenous gene, and then appending a variety of domains that either have activating or repressive activity. The next sort of iteration of this was the tail proteins. So the tails were supposed to be the best thing, and then Cas9 came along. These proteins have a more controllable, or they have a more defined code for recognizing nucleic acid or DNA base pairs. So the tails, just as a reminder, have these 34 amino acid repeats. Then they have this code where you have different combinations of amino acids that recognize specific base pairs. And if you string together a series of these 34 base pair repeats that recognize the desired DNA sequence that you already know what you would like to bind, you can create an artificial protein that very efficiently recognizes specific DNA sequences in a programmable way. And this was sort of more straightforward than the zinc fingers, and people copied more or less what had been done with zinc fingers by fusing activation domains. You'll see this VP64 domain pop up here again, or repression domains. You'll see SID or CRAB domains that can repress expression of endogenous genes. And so the zinc fingers and the tails were, phase one was to figure out how transcription works and use just chimeric fixed sequence transcription factors. Phase two of artificial transcription factor research was the evolution of variable or a programmable sequence transcription factors that you could fuse to domains and turn endogenous genes that are unmodified on and off at will. And people showed that this worked across a series of genes, and sometimes you needed more than one tail to turn genes on, but that it was a general tool that could be used to study transcription. So, like I said, though, the problem with all these methods is that you need to create a new protein for every desired target sequence. And that's not so much that's actually not a problem at all if you have a therapeutic application where you're interested in spending a lot of time defining the activity and specificity of a specific nuclease or transcription factor to have some function. But is a problem for individual labs where they just want to mutate or turn on or off some specific gene, and it's a graduate student and they just want to get to the answer of what the gene does, and so would like to move very quickly. And it's also really a major problem if you want to ask how what the function of every gene is in the genome. So if you want to mutate all 20,000 genes or turn off all 20,000 genes or turn on all 20,000 genes, almost no labs are capable of doing this. There was a TALEN library described as a genome-wide library, but it was enormously expensive. And so really a lot of what I'm talking about today is useful primarily because it's allowed us very quickly to move to genome scale experiments that Martin will talk about later. So turning on and off one gene is fine. Turning on and off all of them is way more interesting. Okay, so after that very long introduction, and we've finally arrived at the CRISPR portion of the talk, which is what you actually all came for. So, but I feel it's important to understand where these tools come from and why we were able to go from Jennifer's paper being published in August of 2012 to less than 2 years later having genome scale libraries for Cas9 for mutating the genome or for turning off or on every gene in the human genome. And that was largely only possible because we knew a lot from RNAi about how to build and use libraries, and then we knew a lot from artificial transcription factors about how to turn genes on and off. All right, so most of this workshop is about Cas9. Like I said at the beginning, at UCSF and influenced early on by Stanley, we've primarily used a nuclease inactive version of Cas9. We view this as an RNA-guided DNA binding platform. The first series of experiments, which were largely done by Stanley, were to show that dCas9 can be expressed in cells and will bind to DNA in a sequence-specific manner, and that it actually does something. And so I don't know actually what Stanley thought he would see when he did these initial experiments. I think, so the experiment he did first was to take the pyogenese Cas9 and express it in E. coli, targeting an RFP reporter. And what he saw was that he recovered almost no E. coli colonies. So in E. coli, if you make a double-stranded break in the genome, it's not repaired efficiently and those cells just die. And so that he now viewed as not that useful of a tool for engineering E. coli genomes where the E. coli genome is already fairly manipulatable. And so genome engineering in E. coli doesn't necessarily need Cas9. But from that, I think he wondered, well, is there something that we can use Cas9 for in E. coli? And so he expressed, I think almost as a control, the nuclease deactivated version of Cas9 just to show that there was no intrinsic Cas9 toxicity or toxicity from the guide RNA, and targeted it to the genome. And what he saw initially was the following. So if you take the pyogenese type 2 CRISPR system that almost everybody is using, make 2 point mutations in dCas9 in the 2 nuclease domains, rendering it catalytically inactive, and then transfer the system to E. coli and express a guide that targets an RFP reporter. If you express Cas9, all the cells are dead. If you express dCas9, what Stanley saw was that depending on where you target these guide RNAs relative to the transcription start site of this RFP reporter, you saw a dramatic decrease in gene expression. So that's shown here, where he saw that if you target these 3 guides to the non-template strand, you get between 10 and 150-fold repression in RFP signals. So he's targeting an RFP reporter, so you can very easily see that it's affecting transcription. And I think this probably initially came as a surprise that there was such a profound effect on transcription if you target Cas9 to the E. coli genome. What I don't have shown on this slide, but was equally interesting, was that if you target Cas9 to the template strand, you see really very, very little effect on transcription. So this is a nice internally controlled experiment where if you target the non-template strand, you turn off transcription. You target the template strand, transcription stays on. And this suggested that, for still completely mysterious reasons, Cas9 is capable of blocking and elongating RNA polymerase if you bind it to the non-template strand, but not the template strand of a gene in E. coli. Together with Matt Larson in Jonathan's lab, he did a preliminary RNA-seq experiment to ask whether the ability to control transcription in E. coli was specific. And so they were able to show that it is quite specific. This is a log 10 scale, so you see that when you target RFP goes down, and it's the strongest effect in the cell. And so this implied both that you could robustly and specifically control transcription in E. coli, and suggested that this could be a useful tool independent of any genome engineering activity from Cas9. So there were a lot of questions about how this actually might work, though, right? Like most proteins, when you bind them to the genome, don't block an elongating RNA polymerase. You can imagine that this might not be something a cell would want to do. And so, again, together with Matt Larson in Jonathan's lab, Stanley set out to ask what was actually happening to RNA polymerase. And fortunately, Jonathan's lab had developed a method called NetSeq, which stands for Nascent Elongating Transcript Sequencing. And this method relies on a tagged RNA polymerase that you can cross-link to. Actually, I don't know if they cross-link. That you can. No, they must cross-link. That you can cross-link your bacteria and then pull down the tagged polymerase and measure where every polymerase is across the genome. And so they did this experiment in the presence of Cas9 and a guide RNA, no guide RNA or a guide RNA targeting this RFP reporter. And what they were able to show is that, so these peaks are counts of where RNA polymerase is along the gene body. And you see that without a guide RNA, you see RNA polymerase transcribing RFP, not completely evenly distributed, but definitely present across the whole gene. And then in stark contrast, if you express a guide RNA which binds right here, you see that RNA polymerase begins transcribing RFP and transcribes along quite happily until it hits the edge of where Cas9 is bound. And you see a peak of RNA polymerase piling up, and you see no RNA polymerase beyond this pile up. So this experiment very clearly illustrated that in a live cell, that Cas9 is binding to a specific region of the genome, and that this binding event, when on the non-template strand, is capable of almost completely blocking and elongating RNA polymerase. Stanley went on to show that Cas9 can block protein interactions with DNA beyond just RNA polymerase. So he was able to show that Cas9 can actually compete with transcription factor binding directly. So if this is a promoter of this RFP reporter, and he was able to show that if you express different guides labeled P1 through 5 that have overlap with different regions known to be important for promoter activity, such as the minus 10 or minus 35 boxes, you see that you can block transcription of RFP. So this was the first indication that we had that Cas9 could directly compete with access to DNA with transcription factor access to DNA. So how am I doing now? This is actually sort of perfect timing. Okay, I'll finish the bacterial portion in the next 3 minutes, and then we'll do eukaryotes tomorrow with no background, I promise. So Stanley he really did a fairly remarkable series of experiments in my mind in which he showed why this might be a useful tool in bacteria. So he showed that gene repression was inducible and reversible. So his initial Cas9 constructs were tetracycline inducible. So if you add tetracycline and look at RFP levels of this reporter, you see their genes well expressed at the start. Add tetracycline, the gene goes off. Wash out tetracycline, gene expression comes right back. So that might be useful for a variety of applications. Gene silencing was also synergistic, so you express two pretty good guides. You get 200 or 100-fold repression. You express both of them together. Now you're up to over 500-fold repression. He also showed that it could be used, that CRISPR could be used to control expression of multiple genes. So here he did an experiment where he's targeting either a GFP reporter or an RFP reporter. You can see that when you target the GFP reporter, GFP expression is turned off, RFP stays on. You target the RFP reporter, RFP goes off, GFP stays on, or you can target both of them. And these are sort of toy experiments, but indicate that you can control the expression of multiple genes, and that this might be useful for a variety of things, such as epistasis analysis or rewiring complex pathways that have branch points for metabolic engineering. And the specificity here is not, the specificity was defined by the RNA-seq experiment, but still this was nice to see. So this paper ended with some sort of systems analysis of the lac operon in bacteria that were indicative that this might be useful generally for understanding pathways. But I wanted to highlight that that things didn't end here in bacteria. So these CRISPR tools are being used in bacteria. I know there's a bunch of unpublished work where people are using these tools in tuberculosis. So that'll be interesting to see come out over the next year or two. There's a nice paper from Carol Gross's lab as well as from Stanley showing that CRISPR is incredibly useful in Bacillus subtilis that just came out. So here, this figure is essentially it's sort of an exact recreation of Stanley's original figure, in which they show that they can construct a xylose-inducible dCas9 system. You get a small amount of leaky repression in the absence of xylose. You add xylose-induced dCas9 expression, you turn gene transcription almost completely off. This is very highly specific. So this is a cleaner RNA-seq experiment where you see that plus or minus a guide, RFP is really the only gene whose expression changes. They did the same NetSeq experiment, again, show that here's RNA polymerase, it's all along the gene body of this RFP reporter. You add a guide, all of a sudden RNA polymerase stops right at the edge of where the Cas9 guide complex is on the gene body. And then they showed again, and still for mysterious reasons, that repression requires in bacteria targeting the non-template strand. So these are like 7 or 8 guides targeting the non-template strand. You can see the transcription is almost completely turned off, where if you target the template strand of the exact same reporter, transcription is either unperturbed or modestly down. So we don't still understand this, but it is what it is. And then I won't go through the whole paper for reasons of time, but what they went on to do is a lot of work looking at antibiotic resistance and showing that they could identify the direct molecular target of a series of antibiotics that where people knew nothing about the way that these antibiotics were working. They did a bunch of chemical genetics. And then they do a lot of work on essential genes. So essential genes are hard to study, right? But now you have a tool where you can, in a titratable way and in a temporal way, turn gene expression on or off. And they ask why, essentially, for all essential genes in subtilis, why are they essential, and what are the terminal phenotypes if you repress an essential gene, which I think hadn't really been done in bacteria. And so with that, I'll end. And tomorrow morning, I'll talk about The rest of the talk is all human. It'll be all about controlling transcription in human cells, imaging the genome, or imaging the transcriptome. And thanks.